Welcome back to the Student Hub Live STEM Showcase. In this session, we have life, health and chemical sciences, and we're splitting this session into two parts. The first part, we're going to do a chromatography experiment, and in the second part, we're going to show you a reality app and look at the human heart. So it's going to be a very exciting session. I welcome Claire Turner and Nick Chatterton here to the studio today. So, chromatography, where and when does this take place, and why is it so interesting?
Would you like to start? Yeah. Okay, so it's used I think it's a key technique in many different sort of areas of science, or forensic science, to identify, sort of the trace amounts of a particular chemical. Also food science to help quality control, and also very much in sort of control of sort of banned substances in terms of like sport and that sort of thing. Because what you're able to do in this technique is identify sort of small amounts of chemicals that could give a sort of a signature as to that they're there and how much is there. So it's really, really important in all kinds of areas, certainly in chemistry, but generally in science. And it's a method of essentially separating chemicals from a mixture, and also sometimes being able to quantify the amount of those chemicals.
Now, we've spoken today a lot about how students do science and do practical science. Is this something that we send students at home, or how might students engage with this sort of technique?
This sort of technique. So in terms of on the chemistry course, we have a practical, sort of screen practical, which involves real sort of scientific data from research papers, where essentially what they're doing is monitoring how different drugs are metabolized, okay? And that can be very important in terms of the safety applications of that kind of thing. So we have, what we do in that situation is we have a lab cast, which is where some of us go into the lab and give a live broadcast to introduce the technique. And also there's a remote aspect to this experiment as well, so that you can connect. We've recently invested in some HPLC, which is more sophisticated than the type I'm going to show you today, type of chromatography experiment, which students eventually will be able to remotely access and carry out this kind of technique themselves.
Brilliant. And we've looked at some of the remote aspects earlier, and a bit later in the programme today, we're going to go and look at how some people are setting up for one of those lab casts tonight.
Okay. So,
are you going to show us then some chromatography and explain how it all happens?
I sure am. So this is actually a demonstration we actually do in one of these lab casts, and this is really just to give us a sense of how the technique works. What we have here is a syringe, which I've sort of plonked into the top of what's called an SPE cartridge. But the key thing about this stuff is that this is white solid column packing material. Okay, and that's that stuff that's going to do the work. Okay, so before we do the actual separation, because what we're actually going to be doing is separating. We've got some food colouring, which is made of a mixture of two different food colours mixed together. We're going to separate into the two parts. But before we do that, what we have to do is actually prepare the column, which will take a few seconds. So I'm just going to run through the first solution. I should put my safety glasses on in a second. Because it might, hopefully won't, but it might go everywhere.
I love the way you haven't bought some for me and Claire. No, we'll be fine, Nick. Should be fine. We're disposable, I think. Yeah.
So you see it's hopefully coming through, and we push that stuff through. Oh, great stuff. So that's the first one that goes through, and that's literally because the solid material is all packed together, and you've got to kind of loosen it up. So we do that by running through two solvents to sort of loosen up the packing material. Is where I have to be a bit careful. Try and keep it all just more rapidly than that. Take that out. Get more air in, and then just push it through. But hopefully this will be more effective. Second time. So I'll just do that again, one more time. So just pushing the solution through, and all I'm trying to do is open up the pores in the solid material, such that the second material, which actually the key one is this second liquid I'm going to pour through here. I won't get into the details, but what it's going to do is add some sort of charge to the solid material, which will help this separation. So I'll add a little bit of that, and it's basically the same again. 3 mils. Try to make it not go everywhere. That's basically what I'm trying to do. So 3 mils. Same idea. Thing in. This is basically just like a sort of a bit like washing up liquid. So you might sometimes see it. You might see it froth if you look carefully. Don't know whether you've got a close up of this.
Yeah, we can see relatively close, but not in too much detail.
So just push that through. So that's the first two. That's now the column prepared, and now what I can do is add the food colouring. Okay, And that's a mixture of two different food colours. And so I'll just do the same with that. Let's get a bit better clamped. Helps it look loose. And 3 mils of that. And what we'll see here, what we're doing in the first step is just trying to concentrate that onto the top of the column such that the separation is more effective. So 3 mils of food colouring, approximately. And then press down again. Again, do this as slowly or as quickly as you wish, but slow is good sometimes. Try and break the airlock. And then remove it, and then push it onto the column material. Oops. Really got any air. So hopefully you might be able to see that that's sort of concentrating sort of darker green colour at the top of the column.
Might be easier, Nick, if you move when you've finished that one away from the green background.
Oh yes, all right, that's good thinking. Okay, so just pushing it through, and then last but not least, do it one more time. And then
I guess a good thing if you're demonstrating this, because add more air might be a difficult
yeah, well,
instruction
exactly.
for students to do at home, but it's an important part of the process.
It's important, but just to push the stuff through, essentially. Although that's what's going to be pushing against the solution,
Yeah.
pushing it onto the solid material.
Yeah.
So hopefully you can now see you've got a sort of a material that's packed onto the top of the column,
Yeah.
and that's the green stuff. And what we're going to do is essentially separate, add two. We've got two solutions that I prepared, hopefully labelled correctly 1 and 2. The first one will separate so basically they're different. They've got different compositions essentially. So this stuff is essentially stuck to the top of the column. Okay, the mixture is stuck to the top of the column material. And what we'll do is push through these two liquids. And then the way the process works is that one of the two food colourings in this mixture will have a greater affinity for this one. So it will separate that out. You'll see hopefully that the first food colour comes off. So I'll just go ahead and do that. So we've got solution 1, again approximately 3 mils. Put it through, and then slowly but surely push that through. Okay. There we go. And then we should really switch these over, so I will. So you can collect the solution that we want that we're interested in. So hopefully, if you can see, you're getting that nice separation at the bottom there. There's the blue colour, the blue solution is being pulled off, and the other stuff, which you may be able to see, the yellow stuff, is staying attached to the column. So you can push that through really neatly. So we're separating the blue component of the mixture, yet leaving the yellow component behind.
And we can see the blue in the bottom as well.
And the blue, we're collecting, we're not 100% pure, but a purer version of the blue with very little of the yellow remaining. So I'll just finish it off, just push the rest of that solution through, hopefully. Don't seem to be too much there. So again, I'll need a bit more air, just to get it off before I use the final solution. And then just keep going. It's coming off quite slowly. Just keep going, collect the rest of the blue, and that will be good. So hopefully you can now see that most of the blue, or virtually all of the blue has come off, and the yellow has really remained stuck to the column. So what we're now finally going to do is change the solution again to solution number 2, and use that to pull off the yellow. And what we've essentially done through this whole process is separate the yellow from the blue food dye. So let's just finish this off. Hopefully this is indeed the right mixture.
Now, while you're doing that, Nick, Claire, I wonder if you could tell us how this links to research.
So chromatography is one of the kind of really fundamental techniques in much of scientific research, because when you have a whole pile of different compounds all together, as you often will do, you do need to separate them before you can actually analyse them. So the kind of research that I do is I'm interested in diagnosing disease, actually non-invasively, and pretty much want to do this by trying to find biomarkers for particular diseases. And the thing with that is if you take a sample from a human, so if you take a sample of urine or blood or any of those things, as you can imagine, they have got lots and lots of things in them. So if you want to try and pick out what those biomarkers are, which are actually, indicate what the disease is and what have you, you really do need to separate them. So chromatography is absolutely a key technique to use to do that, and it goes across all sorts of different areas. Now, Nick's just really very nicely demonstrated that, because you can see the yellows come out very clearly.
Yeah, so you've got the yellow.
So he's very nicely demonstrated that you've got...
You can put them on the table, we should be able to see them against the white tablecloth.
You can separate two compounds, but in actual fact, in some of the techniques, which are a lot more bigger and expensive and what have you, but they can actually separate thousands of compounds in a sample. So this becomes a very, very powerful technique. And as you separate each compound, as each compound comes off, you can analyse it. So the analysis techniques, much easier to analyse a compound if it's one compound on its own rather than thousands of them. So.
And this is something the students will meet in level 3 in the lab casts.
Absolutely, because in terms of this is to introduce the phenomenon that they're going to use to analyse the more sophisticated, as Claire's mentioned, types of instrument that you actually be using, just so they understand how the solvent plays a role, the solution plays a role, and how that, because it's basically interactions between the solvent molecules, which I push through, and the dye that's given rise to the separation. So they need to understand a little bit about how that phenomenon works. So we just demonstrate it using this sort of type of demonstration.
Excellent. Well, thank you both so much for coming and showing us that. That's been absolutely excellent. So Nick and Claire, thank you.
Thank you very much.
While we're preparing for our next session, let's just take a quick trip to the hot desk where we have Eleanor, who is from the Life Health Science, see, I'm going to get it wrong now, but I'm going to say it, Life Health and Chemical Sciences team, and she is joined by Sarah on the hot desk. How is everyone back at home? I imagine everyone was watching that quite avidly.
Yeah, very interesting conversations with some of the students.
So there's discussions on S111, which is the first module within the science. They actually do quite a lot of kind of kitchen chemistry, and one of the experiments they do there is separating kind of marker pens using kind of like coffee filter paper. So there's kind of discussion about that, which I think has also taken some people back to their school days when you'd have done something quite similar. So.
We've talked a bit about kitchen science as well, and how that's integrated, and there's some quite interesting experiments as well in S111 out there that people can do at home.
Well, we should have. Nick would have been the ideal person to talk about that, because he's actually on the 111 module team, so he would know about all the different types of experiments that there are on the module. Yeah, so a whole range. I think they'll be right across the because S111 covers all the different sciences, so there's a whole range of different experiments that are there.
Brilliant. And Sarah, how's everyone back home? Everyone think about afternoon tea yet?
No, we've had no conversations about cake or anything. I'm quite disappointed.
We've got discussions about gin and tonic and things, haven't we?
Oh, who's that? Because I was talking about gin and tonic
that one's just popped up. I think we had a discussion. Somebody was talking about kitchen science, and they were talking about, I think Davin was talking about making pigs in blanket toad and hole.
Oh, it started now, the baking's all coming through.
And someone was wondering if you could do experiments like this to separate gin from tonic.
You should never separate gin from tonic. I think it's a very bad idea.
I've got another, and Carol's just fed in here from S111, was one very successful experiment that she had was to extract DNA from kiwi fruit.
Oh. Yes, I've heard that was a good one.
But I shall leave that for your next.
Brilliant. Well, thank you very much, Linda Cook, who's returned today. What have you been doing out on campus today, Linda? You came in for our introduction earlier.
I did. Well, we've been checking to make sure I got everything ready for this session this afternoon. So, it's quite an exciting day.
Excellent. And welcome Martin Beekman to the session as well.
Hi.
Now, you're going to show us a reality app, which is really exciting and completely different to what we were looking at earlier. This is about the human heart.
It is. So our introductory sort of second level module called Human Biology for biology or health students. One of the topics is cardiovascular physiology, and of course the heart is a key component of the cardiovascular system. And it's really important. The heart beats from 16 days post-fertilisation right through our life all the time. To understand the sort of how that structure is related to the function, we can use various things like diagrams, or we can use models like we've got here as well. But how our students actually get to grips with it, you know, we can get them to label things and things like that, but actually using a reality app like we've got here, they can actually see it moving and they can interact with it and learn how the structure is related to its function.
It saves all the mess of dissection as well.
Oh, yes, absolutely.
Yes, which I was never very keen on.
So I don't know whether I can show this on
Yes,
camera
please
or
do.
not.
So if you just hold it up so that we can take a look at this. So this is the app, and we're going to show it on the screen as well to take a look.
Right, so this is available it's actually available to anybody on Google and Android as well. It's called SK277 Heart app. And what happens is you download it. You can download it as a desktop version or as a tablet version here, as you've shown here, and you can rotate it round. Okay, so it's an anatomically accurate model, and we have a menu that comes up here, and we can do various things. So we can open the heart up, so we can actually see inside it. If we get with the menu, you can see that a bit more clearly. Okay, so you can see the 2 upper chambers and you can see the 2 lower chambers. Okay, so that's quite similar to students using a diagram, but what we can also do is we can actually play the heart as well, and we can get rid of that. And students can do this, they can do that at their own pace. And we can guide them in the module materials with certain questions to make them investigate aspects of the heart and how it functions. Okay, so you mentioned about dissection. Well, we can have some pins, dissection pins. So if I just stop the heart beating so you can see this a little bit more clearly, I can get rid of that. And then you can press the pin and it comes up there with the label.
Brilliant.
So what's really important to understand about heart function is the coordinated contraction of the upper chambers, moving blood flow into the lower chambers and ejection, and that's got to happen all the time to keep us alive. Now we can also show blood flow as well, so I'll get rid of that. So students can do that, and they can turn the app around. Let me just make it a little bit more smaller. They can see the valves closing and opening. Okay, so, and they can also link that, as I say, to important biological sort of processes and physiological processes that are going on. So we have here what's called the cardiac cycle. And if I just move this along here, whoops, can't do this, so you can see it. Yeah. If I can get it to be wrong, okay. So, and it's really important that students grasp what's called the cardiac cycle. That's one, each heartbeat, what is taking place, and it's actually quite complex. So this gives a really good opportunity for students to gain a lot more from a stationary diagram. They can really get into the function of the heart and look at it, and as I say, doing it at their own level, and we give them learning opportunities to engage with this so that they can develop their own learning.
What an amazing way of studying, because so often, I mean, you know, you can have models, you can read about things in textbooks, but one of the things that I often find, you know, my students struggle with in terms of biology is that you'll read a certain thing and then read a certain similar thing but from a different perspective or looking at a different plane, and it can make it really hard to put things together, to conceptualise them, especially when you've got text and diagrams. And we have a lot of, you know, images and, you know, multimedia things that really help conceptualise. But this is really different, and being able to control it and look around creates just something so unique. I can't believe it's free to everybody.
It is, yeah, and we've got, it's Paul Hogan from our KMI has developed this, and as I say, it's available on the App Store and on Google for Android as well.
And
but
KMI are our Knowledge Media Institute who develop all sorts of fabulous things, including the Stadium Live interface.
Yeah. So it should, and I say we have activities that students engage with this, and then that's part of their assessment as well. So we make sure that, you know, that they can test their understanding as well.
So what sorts of things would students be doing then from looking at this? These will be activities within the module, and which module is that?
So this is SK299, which is human biology. And we would ask them to actually be able to understand a cardiac cycle, all the components of the cardiac cycle, which you saw in that quite complex graph that was shown on there. And this would enable them to do that. And actually, it links to our research, and it's in terms of understanding how the cardiac cycle, how each cardiac cycle takes place, links to each heartbeat and how it functions.
Excellent. Did you want to show us anything on the monitor as well with this, or have we covered most of the functions?
So actually, Linda's mentioned the cardiac cycle a couple of times, And the cardiac cycle is driven by the generation of electrical signals within the heart, which the app really nicely shows. And we've had a long-standing expertise in cardiovascular biology here at the Open University, and indeed we develop assets like this, but also movies that you alluded to earlier. And one of the things we've been trying to understand for many years is this cardiac cycle and what happens when it goes wrong. And what we've brought with us today is a couple of movies to show you this kind of cardiac cycle as it functions in a kind of a beating cell. So if I play this movie, let me just go back and find the mouse. Here we go. And I just play this. Now what you can see here, let's just get rid of the mouse. If it's playing on the screen, is it going to play?
I think it might have paused. You might need to hover and press the play button again.
Okay, let me try that again.
Yeah.
There we go. So what you can see here is a single beating heart cell. And the change in colour that you can see is actually a surge of calcium within the cell. So the heart relies on electrical signals to coordinate its beating, and so it operates like a pump. It's a very strong muscular pump. But when those electrical signals reach each individual cardiac myocyte, as we call the heart cells, what happens is that there's a surge in their calcium concentration, and that surge in calcium concentration causes them to contract, and you can see that cell contracting just from that edge moving in. And of course, that's a nice rhythmic pattern. That's what you want. That's about the speed that most human hearts go. Mine's going a little faster right now. But sometimes it goes wrong. And this is an example of when it goes wrong. What you can see here, in this case, is two heart cells beating away. I'll just wait for that to load up. Let me try that again. You can see two heart cells here. Okay. And they're beating away, and the top cell stays faithful to the rhythm that we want. There's an electrical signal imposed on it, but the bottom cell starts to do its own thing. And you can see it started to skip beats. And eventually, about now, it stops beating at all. And it stops showing those calcium signals. And for a long time now, we've been trying to investigate what causes that change. Why does it go from a rhythmic pattern, which you need for the heart to act as a coordinated pump, to squish the blood to the lungs or to the body? Why does the heart fail? And one of the things that we know that we have studied for some time is that the heart cells are this precisely geometrically aligned cell type. You can see here, this is an image of a heart cell, and it's what we're showing here in this image is one particular protein that's expressed inside heart cells. It's called ranitidine receptors, which are part of this calcium signaling machinery. And you can see these are beautifully striated cells. We call these line pattern striations. But as we age, things change. And here at the Open University, we've got a fantastic electron microscopy suite. And we can look at the ultrastructure of these cells and see what happens as we age. On the left, you can see just a small representation from a healthy heart cell, from a young heart cell. And you can see these beautiful striations again. And this is how the cells need to be set up so that they can contract maximally and our hearts can work really nicely. On the right-hand side, you can see a representation from an old heart cell, and it looks completely different. And we don't fully understand that change, but we know when that change happens, the heart isn't going to be such an effective pump, and we're going to be poorly. And that's what we're researching right now. And we use our expertise to feed back into courses such as 299 to make sure that we're informing students about really cutting edge results and experiments.
We've talked about how a lot of the curriculum extends beyond the curriculum and research students, and you say we're researching. Yes. Who and what opportunities might there be for students in the future to do important work like this?
Well, indeed, these particular images here were actually obtained by a PhD student in my lab, Saeed, and he's just actually finished his PhD. He was very successful, had a very enjoyable viva, and he did actually spend his 3 years doing research with us, investigating the changes that happen in the heart during aging, which is extremely topical. We have an aging population, as you know. We need to know why do people's hearts become less effective as they age? Why do they develop symptoms such as hypertrophy and arrhythmias and things like that? So we have lots of opportunities. We, as well as PhD students, we try to get our undergraduate students involved as much as we can. We have internships. We have a lot of visitors that come, and we're very keen to disseminate our research expertise. We go off to conferences. We publish a lot of papers, and we do engage where we can And certainly, we have a conference next week, in fact, actually dealing with some of these issues, and that's going to be available on Stadium. We really want our students to feel part of our research community and to understand the research we're doing.
And where can students find out about that?
So that's available on the Stadium webpage and also on the Qual's website for
science. Well,
science.
hopefully we'll put on the,
Yes,
on the science
yes, it is.
study website.
Yes.
Brilliant.
So-
But if you are interested and you drop us an email, then we can always email you the exact link a little bit later this evening. Well, that's so interesting. These PhDs then, I mean, it seems like a real area of concern. Are a lot of these predefined and students will apply to, for example, pick up a specific topic that the team are researching? Or can students come in with their own ideas and say, this is something, could the Open University consider letting me do a PhD here?
Well, it's always a bit of a mix, actually, and very often we have students that have been really engaged with a topic during their undergraduate study or perhaps a master's study, and they come along and they have a real passion. And when somebody has a passion, then you've got that motivation that you need to see something like a PhD through successfully. So, but we also have lots and lots of projects that we want to undertake. So we're very keen for anybody who thinks that research and understanding, making fundamental discoveries. And it can be quite taxing. I mean, Linda and I both gone through PhDs some years ago, and we know that experiments don't always work, and we think we understand how cells behave, but they have a habit of being much cleverer than we are and understanding. We have a lack of understanding of things that sometimes we think we got to grips with, and that's why research takes such a long time. That's why the development of pharmacology and new therapeutics takes such a long time, because cells are actually incredibly complex computing machines.
And of course the environment's changing because, as you say, with an ageing population there are different things that are being thrown up all the time, and so it's continuously relevant and evolving.
Yes. Absolutely so. Yes, I mean, the missions from research councils and from funding bodies change over time. The ageing is extremely topical right now and will continue to be as our population does age, but of course there's an enormous emphasis on conditions such as asthma and hypertension, and obesity and diabetes. These are very important societal issues right now, and there's a lot of funding. So and new challenges will arise.
Excellent. Well, thank you both so much for coming along today. It's been incredibly interesting. I just want to take a quick trip to the hot desk. Eleanor, anything else that we need to mention before we end this session?
I think there was some discussion about where we might find the app, so it was kind of emphasising that's on the Open STEM Labs website, and just checking, I think that it is available to everybody. I think Linda said that, yes, so it's available to everybody. I think that was one of the main discussion, other than the fact that the gin is rumbling on. And there's some nice recipes on there as well, for people to try over Christmas. But yes,
I shall look forward to catching up on those later. Well done, everybody at home. Okay, well, thank you very much for chatting with us. We're going to take a short video break and show you about the MSc in mental health, and then we're going to meet more researchers where we're going to look at waste management, recycling and plastic. So join me then in 5 minutes.